Hey guys, my name is Joey and I have a bachelor's degree in biology with a minor in chemistry, and my passion is science. Once a week I'll create a video that discusses mangas such as One Piece, Hunter x Hunter, and Gintama. I will also be discussing scientific articles that I have been reading and connecting them to other articles and books. They may not be the most popular or scientific trending articles out there. Lastly, I'll be discussing policies that are taking place throughout the world. They may be in the past or present. So, BBC published an article reviewing a study published in Nature that examined all the potential errors that DNA in breast tissue may undergo to produce cancer. The goal was to understand how the DNA in healthy tissue can become cancerous at a molecular level. The team looked at the genome of 560 breast cancer patients and discovered 93 genes that, when mutated, can cause tumors. Scientists have now learned which genes will cause breast cancer. The question then becomes, how is this beneficial, and why is this such a milestone? Professor Sir Mike Stratton, the leader of the study, said the list of the 93 genes can be given to universities, pharmaceutical companies, and biotech companies to start developing new drugs that will be able to target the mutated genes and the protein they produce. Back in the 1970s, scientists discovered that miscoded genes can lead to cancer. These genes can be called oncogenes. As it was discovered that cancer is the result of genetic mutations, scientists decided to create drugs that would target the cancerous genes and their products. One great example is the Philadelphia Chromosome. Each of your chromosomes is divided into two pairs, one given from your father and one given from your mother. Thus, there are a total of 22 autosome pairs and one pair of sex chromosomes, or a total of 23 pairs of chromosomes. Sometimes a chromosome will rearrange with a different chromosome to create a new and fused chromosome. As you can see in the picture, the bottom of chromosome number 4 is replaced with the bottom of chromosome 20. This is known as reciprocal translocation. In the case of the Philadelphia chromosome, chromosomes 9 and 22 will go through a reciprocal translocation to produce a protein that has a combined structure of two different genes. In chromosome 9, there is the ABL, and in chromosome 22, it's the BCL. The cells that are affected are the white blood cells, and thus it leads a person to develop chronic myeloid leukemia. The combined structure forces the protein to be continuously switched on, and so the protein will always remain constantly active. As it is active, it will cause the cell to reproduce over and over and over. After many years of research on understanding the protein structure, scientists wanted to be able to create a drug that would target the gene without targeting the rest of the body, as is with the case of chemotherapy. Researchers and doctors teamed up with the pharmaceutical company Novartis to create such a drug. Scientists were able to produce a drug that only killed the cancer cells of animals that had the Philadelphia chromosome. But Novartis was extremely hesitant to begin clinical trials on a drug that would need to make over $100 million in sales just to break even. After much push and 7 years of debating, the company began trials, and the drug Gleevec was the first drug produced to target cells with the cancerous gene and destroy them without harming the rest of the body. Professor Mike Stratton and his team are hoping that by identifying these 93 genes that are able to cause cancer, that they will be able to send the list to pharmaceutical companies so that they will be able to do the exact same thing as with the Philadelphia chromosome. The idea is that by identifying these 93 genes, they can create drugs that will be able to attack each cancer cell with the gene that is of target. There is also many obstacles that need to be overcome before this can be a success. One is that it's going to take at least a decade for any of these drugs to be prepared and ready for consumers, as stated by Professor Mike Stratton. Sadly, there are mutations that are so rare, going to be very few companies that will be willing to invest the amount of money to benefit such a small and tiny population. I'm going to read a quote from the novel Better, A Surgeon's Notes on Performance. To be sure, we need innovations to expand our knowledge in therapies, whether for cystic fibrosis, childhood lymphoma, or heart disease, or any other of the countless ways in which the human body fails. But we have not effectively used the abilities science has already given us. We have not made remotely adequate efforts to change that. When we've made a science of performance, however, thousands of lives have been saved. Indeed, the scientific effort to improve performance in medicine, an effort at present only gets a minuscule portion of the scientific budget, can arguably save more lives in the next decade than bench science, more lives than research on the genome, stem cell therapy, cancer vaccines, and all other laboratory work that we hear about in the news. The point the author is making is that perhaps we shouldn't invest so much money on the newest technologies that the news is always trying to tell us that will save our lives, but perhaps research and medicine should invest more money on improving the efficiency of current technologies that we already have, such as mammograms. One more quote from the author about breast cancer. Rates of death from breast cancer have fallen about 25% in industrialized countries since 1990. A study of data from the U.S. Breast Cancer Registry recently showed that at least a quarter, and likely more than half, of the decline was due simply to an increased use of screening mammograph by women. Mammographs save lives by allowing breast cancer to be caught and treated while they're still small, before they can even be felt, and hopefully before they have spread. But the key lies in working with the women to faithfully get a mammogram once a year. Less often leaves too much time in between for breast cancer to form, grow, and spread undetected. So I've shown you 3 different works from 3 different authors about their views of cancer. And I hope that this has been very educational and that you've learned more about cancer, types of therapies that are being used for cancer. And please keep watching my videos. Please subscribe to me. I'm going to keep doing these scientific articles, and I really enjoy them. And the next one's going to be really exciting. It's going to be about genetics and eugenics. And I'm really excited for that one. That one's going to be a bigger one. It's going to be very interesting. I learned a lot about eugenics that I hadn't known before, and I'm really excited for it. So please stay tuned. Lastly, here are the references on which the information I talked about. You can look at the books. I highly recommend them. And then also here are the photograph links in case you need them or to give credit to make sure that that is received. Thank you very much.